Hi, myself Tul Zuhra, and I'm going to present a research article taken from a signal transduction and targeted therapy. And the article named as COVID-19-associated monocytic encephalitis, histological and proteomic evidence from the autopsy. Now we will discuss the background and further we will go into the detail of findings of this paper. And COVID-19 is an infectious disease that is caused by a severe respiratory syndrome coronavirus 2 SARS-CoV-2. And the normal symptoms of the COVID-19 includes the cough, fever, difficult in breathing, and difficult of breathing. But the different reports have seen that there are some neurological pathologies that are reported in the COVID-19 patient, and the COVID-19 patient and the neurological pathophysiologies include that anxiety, depression, vision changes, impaired mobility, numbness in extremities, tremor, migraines, and as impaired mobility, and many more. But previous reports of neuropathological changes in the brain of COVID-19 patients include microglial nodules, neurophagocytosis, and neural injury and endothelitis. Previous studies also revealed that the possible infection by the SARS-CoV-2 virus in the brain tissue of COVID-19 patients. The neurotropism of the SARS-CoV-2 virus has been confirmed in mouse and organoid models. As see why how the SARS-CoV-2 gets an entry into the brain of the patient, brain of a person, then there are the two routes. One is the hematological route, and other one is the retrograde axonal route. Retrograde axonal route involves the olfactory bulb and the enteric respiratory to the blood. If we consider the hematological route, then we will see that there are the different types of the receptors, including the ACE2 receptors and transmembrane protein serine receptors on the COVID-19 patient, or normally that is present on the central nervous system. Then what happened that the spike proteins of the COVID-19 of the SARS-CoV-2 binds to that protein and take their entry into the central nervous system. And once they get entry into the central nervous system, they causes the inflammation or provoke the inflammational signals. Firstly, the different ways by which they provoke or they causes the extremities of the inflammations involved. First one is the endothelial cell, and what happened that the virus particles that is present in the blood get entry into the central nervous system and cause inflammation. Second is the accumulation or the infiltration of the leukocytes into the central nervous system. Third, the pro-inflammatory cytokines that is present in the blood passes through the blood-brain barrier and gets entry into the central nervous system. When there is an increase in the pro-inflammatory cytokines, what happens that they causes that blood-brain barrier dysfunction or the loss the integrity of the blood-brain barrier. What happens when SARS-CoV-2 gets into the perivascular space? They cause and they activates that the astrocytes or the microglial cells that are normally in their inactive state. What they cause that they activate these cells and increase and causes an increase in the inflammatory cytokines. And these inflammatory cytokines causes the inflammation. Pro-inflammatory cytokine causes the inflammation of the central nervous system or the perivascular space that is called the encephalitis. And it has been reported that neither the SARS-CoV-2 messenger RNA or protein is reported into the brain, but there is an increase in the inflammatory responses that are inflammatory molecules or the inflammatory cytokines means there is an increase in their numbers in the patient of COVID-19 and brain. They have evident that result by their autopsy or the different research. For next, we will see the objectives. To better understand the molecular mechanisms that underlie the brain damage, and the second is the monocyte infiltrations and microglial activation as a potential therapeutic target in the COVID-19 brain. Now we will discuss the result. The figure 1, monocyte infiltration and extensive microglial activation in COVID-19 brain. Panel A. The histology shows the neurophagia, microglial nodules, and perivascular inflammation in the COVID-19 brain tissues. Here, in panel A, the first figure shows that there is an increase in the neurophagia, and the black arrows shows that there is an increase in neurophagia in response. And the figure B shows that there is an increase in the microglial nodules. And in third, if we see the third and fourth figures, they shows that there is an increase in the inflammation of the perivascular tissues. And further, we see the figure B, the figure B, the immunochemical staining of IBA1 shows the reactive activation of the microglial, and this area is further elaborated and increased there. Uh, and this area is further elaborated and enhanced. And there can be seen that there is an increase in there is an increase in the activation of the microglia that is the main cause of the inflammation and the encephalitis in the COVID-19 brain. In the figure C, the immunohistochemistry shows that the CD16 and CD14 are positively associated or present in the perivascular tissues infla- uh, in perivascular space and the parenchyma, and these two figures shows that their presence of the inflammatory cells infiltrating in the parenchyma cell. And this area is further elaborated and enhanced by in seeing that there is the presence of the CD14, CD16 in parenchyma cells, and CD14 is also seen in the parenchyma. Further, we will see the CD14 and CD16 presence in the perivascular space, and these hysterics shows the position of the perivascular space in these two figures. Next, the D panel shows the quantitative analysis of the immune cells that is present in different brain tissues. And here we taken the different brain tissues, then that is represented by the different colors, such as the parietal lobe is represented by the gray color, parental lobe by the red color, and as that all, as seen in all the tissues. And this heat map shows the intensity of these immune cells that is expressed in the different tissues of the brain. And the left column of heat map shows the immune cells or represents the immune cells, whereas the right column shows the different types of the tissues that are taken from the brain. If we can see, and this color scale shows the intensity of the protein, or means the immune cells that are expressed in these tissues. Or the gray color shows that there is an absence of the immune cells in these tissues. If we consider this cell IBA1 cell, it has been seen that these IBA cells are more pronounced or more expressed in the brainstem and temporal lobe as compared to in the COVID-19 brain as compared to the control brain. So the color intensity shows their increased expression in the COVID-19 brain and as compared to the control brain. If we see that there is an increase in the number of these immune cells in the different brain tissues, it means there is an increase in the inflammatory responses. If we see that on the heat map x-axis, they are the 9 COVID-19 brains, and they are the 9 control brains. The figure shows the histochemistry of the HLDR, CCR7, CD141, and CD11C. They show that these cells are the monocyte biomarkers are positive for the perivascular space and the perivascular space and the mononuclear cells. And these black arrows that shows the position of the mononuclear cells and the hysteric shows the microvessels of the perivascular space. Further, if further, they perform the proteomic analysis, and in figure F, they show the scheme of the proteomic study that from the temporal lobe, they taken the sample, and what they do that they devaccine protein and peptide digestion. Then they perform the quantitative proteomics, and then they analyze their results by using the different bioinformatic analysis. And in figures G, what they do, they perform the protein component analysis. They differentiated the two groups. That one group that is represented by the red color is the COVID-19 brain, and the blue that is the another group of the control. They perform the proteomic analysis. For this, they taken the sample of the 15 patients. That is the 7 of the COVID-19 brain and 8 from the control brain due to the decrease in the quality of the results. They excluded the one control and 2 COVID-19 brain patients. And they further performed the proteomic analysis with the 15 samples. What did see that there are the 4587 proteins were identified in the COVID-19 patients and 4414 in the control brain. And then they perform the principal component analysis to identify the differentially expressed proteins in the COVID-19 brain and the control brains. And they have seen that the 572 proteins were significantly expressed in the COVID-19 brain and control brain. They further perform, see in the H graph, they further perform the volcano plot, and they see the different types of the proteins that are significantly upregulated and downregulated in the COVID-19 brains and the control brain. And they see that 294 proteins are significantly upregulated in the COVID-19 brain as two folds from the from the control brain. And in the volcano plot, the significantly upregulated proteins are expressed by the red color, and they have also observed that about the 278 proteins are downregulated in the COVID-19 brain as half of the control brains, half fold of the control brains, and these half fold that is significantly downregulated are expressed in the blue color. And they further perform the enrichment, the pathway analysis, and they have seen that the different pathways, including the JAK signaling up in interleukin 12 stimulations and AMP2 neutrophils pathway, and also the monocyte aggregations, these pathways are upregulated in the COVID-19 patient when compared to the control. Now the figure 2. For the further evaluation of the inflammation, they investigated the structural damage and the molecular alternation of the blood-brain barrier in COVID-19 brain. In figure 2, the panel A shows that the hematoxylin and the eosin strain shows that there is an increase in the hyperemia and the dilation of the blood vessel, and also there is an increase in the perivascular infiltration of the inflammatory cell in the blood-brain barrier. This 50 micrometer shows the scale bar. Now, in this figure, the glial fibrillary acidic protein shows that there is a destructive and the damaged glial membrane, and this destruction is shown here. That this semicircle shows that there is a destruction in the glial membrane. If we see the panel B, they shows that the electron microscopy shows the destructive blood-brain barrier and also reveals that the swollen glial membrane and broken basal membrane and the degenerative endothelial membrane, and there is a abnormal tight junction in the blood-brain barrier and and degeneration of the mitochondria. If we see the panel C, in panel C, the histogram shows the relation or the perivascular space in COVID-19 brain or the control brain. We see that the x-axis shows the different tissues of the brain, brain stem, cerebrum, and the occipital lobe and the parietal lobe. And if we see the y-axis, then it shows the area or the area of the perivascular space in millimeter per square. This histogram shows that there is a significant increase in the perivascular space in COVID-19 brain when compared with the control brain. And these hysterics shows the significant values. As the hysteric as the hysterics increases, it shows that there is a significant relation between the increase in the perivascular space in these tissues. The panel D shows the gray cortex glial fibrillary acidic protein staining area in different tissues of the brain. In x-axis, the different tissues of the brain are illustrated as the brainstem, parietal lobe, and hippocampus, and temporal lobe, cerebrum, frontal lobe, and occipital lobe. And in y-axis, the GFAP staining area in percentage. And they observed and shown by the histogram that there is a significant increase in the GFAP staining area in COVID-19 brain and as compared to the control brain. As we know that the GFAP is used as a marker for the astrocyte activations, and when there is an increase in the astrocyte activation, that means that there is an increase in the inflammation in COVID-19 brain. In panel E, this shows the correlation between the perivascular space on x-axis and the gray cortex glial fibrillary acidic protein staining area in y-axis. This shows that there is a significant correlation between these two factors, and they see that the high correlated area of the brain is the brain stem. This shows the correlation of about the 0.89, means the 89% of the correlation exists between the perivascular space and the gray cortex glial fibrillary acidic protein staining area. It shows the significant red. They further performed the proteomic analysis to investigate the damage on the blood-brain barrier. In F figure, the heat map shows the intensity of the different extracellular matrix proteins that is expressed, and the column on the left of the heat map shows the different types of the extracellular proteins, and the x-axis. On x-axis, this shows the proteins that is present in the basement membrane. It has been shown that these proteins are very less expressed in COVID-19 brain as compared to the control brain when they see by their intensity of the color when comparing by it to the intensity color bar, that this blue color shows that these proteins are down regulated in COVID-19 brain as compared to the control brain. Further perform the pathway enrichment analysis, and they shows that the different pathways, including the VEGF signaling pathways that are upregulated in the COVID-19 brain, and different pathways, including the beta-catenin independent WNT signaling pathways and the signaling by hedgehog pathway, they are downregulated in the COVID-19 patient brain. In figure H, they further perform the functional analysis, and this heat map shows the intensity of the proteins that is expressed in different or regulated in different functions. And the left panel on the heat map, left column on the heat map shows the different functions, and the right column on heat map shows the name of the genes. And there are the two different group. One is the COVID-19 brain group, and other is the control brain. And the blue, red, and gray boxes shows the intensity of the proteins that are expressed in these functions. If we see that these red colors, that these protein, that is the TUBB1, is highly expressed in COVID-19 brain as compared to the control brain. And if we see that the TJP2 is highly expressed in the control brain as compared to the COVID-19 brain. And these results, as we see that on the right side is the histogram, and they shows this intensity of the proteins that is expressed in terms of the histogram. This histogram shows that some proteins that are highly expressed or upregulated in the COVID-19 brain as compared to the other proteins that are downregulated in COVID-19 brain when comparing to the control brain. Now the figure 3. This is a similar cytokine profile in COVID-19 associated encephalitis and SARS-CoV-2, lung and liver injury. The panel A shows the heat map, and if we see that on the left column there are different pro-inflammatory inflammatory signals, and on the left side they are the name of the genes that are expressed in differentially in COVID-19 brain and the control brain. When these type of genes sure are their expression is measured, and they show that these type of the pro-inflammatory genes that are involved in the pro-inflammatory responses that are highly expressed in COVID-19 brain as compared to the control brain. These the blue, red, and gray boxes show the intensity of these protein expression in these different signals. And the color red, there is a high intensity of the color that shows that this protein is highly expressed in COVID-19 brain when comparing to the control brain. And if we see that the protein that is the SOD2 protein, there shows that there is an increase in the expression of this protein in the control brain when comparing to the COVID-19 brain. And on the right of the heat map, there is a histogram on the x-axis. They show the log to COVID-19 brain ratio to the control brain, and they shows that there is a increase, or they are upregulated different proteins in the COVID-19 brain, and there are some proteins that are less expressed in the control brain. In panel B, they show that the immunohistochemical slides, they show that the interleukin-4, interleukin-6, and interleukin-8, interleukin-12, and TGF-beta and tumor necrosis factor alpha are differentially expressed in the monocytes, lymphocytes, and the endothelial cells of the COVID-19 associated encephalitis or the COVID-19 associated brain. In figure C, they show similarity of inflammatory factors in lungs and liver and brain of the brain tissue of the COVID-19 patient. And they see that there is a high expression of the pro-inflammatory molecules, that is the interleukin-1, interleukin-2, interleukin-3, and interleukin-12 and tumor necrosis factors. They are highly expressed in the brain, liver, and the lung tissues of the COVID-19 patient. Now the figure 4. Along with the infiltration of the pro-inflammatory molecules of the cells and the damage to the blood-brain barrier, they also observe that there are some different types of the non-inflammatory responses that are responsible for the neurodegenerative disorders or the neuropathologies in the COVID-19 brain. And further, they investigated the neuron and myelin sheath damage associated proteins in the COVID-19 brain. In the figure 4, the panel A shows the immunohistochemistry slides. In the first slide, they shows that there is an increase in the red degeneration of neurons. And in second figure, This shows that there is an increase in the production of the large granular neurons. And in the third slide, they shows that there is then this appearance of the frenal bodies in the neurons. Here, if we see that the panel B depicts the histochemistry of the Purkinje's and the granular cells, and there is an increase in these cells, and there is a decrease in these cells, and their axons and dendrites are injured. If we further see in the C panels, they shows the immunohistochemistry of the myelin sheaths, and they show that the myelin sheaths are swollen and lost in COVID-19 brain. And right image shows the electron microscopy of myelin sheath stained with the Picot stain specific for the myelin sheath. And now the panel D shows the electron microscopic picture of neurons describing the swollen axons and the myelin sheath in the COVID-19 patients when compared with the control. Next, the panel E. Panel E illustrates the signal pathway analysis, and they shows that there are the different pathways on the that are shown on the left of the histogram, and including the nervous system development and the nervous system protection pathways that they are downregulated in the COVID-19 patient, that is expressed by the histogram. And the length of the histogram shows the frequency of the frequency of the downregulation of the different pathways in the COVID-19 brain. Further, they perform the functional analysis, and this heat map shows the intensity of the different proteins that are involved in the different functions. The right column on the left column, the left column of the heat map shows the functions that including the neuronal functions, synapses, and neurorexins, and the right of the column of the heat map are the name of the genes. The red and blue and gray boxes shows the intensity of the proteins that are expressed in the COVID-19 and control. The red color shows that there is an increase in the expression of the protein in and whereas the blue color shows the decrease in the intensity of the intensity of the protein expression, whereas the gray color shows that there is an absence of the protein. If we compare these proteins in control and COVID, it has been seen that these proteins are downregulated in COVID-19 brain, and they are highly expressed in the control brain. On the right of the heat map, there is a histogram that shows the ratio of intensity of the proteins that are expressed. On x-axis, they shows the log to COVID-19 brain to ratio to the control brain, and they assume that these proteins are highly expressed in control as compared to the COVID. So the ratio of or the expression of this protein in COVID are downregulated. In F figure, they illustrate the interaction network that are differentially expressed in COVID-19 or control brain. There are the multiple molecules that are involved in neuropathologies, like many of the protein of the autophagy and the energy metabolism. Associated proteins are highly expressed in the COVID-19 brain. These molecules are interconnected with each other. So from this information, we can assume or make a suggestion that the autophagy process might be responsible for the energy metabolism in the COVID-19 brain. The color bar shows the intensity of these molecules expression in the COVID-19 brain compared to the control brain. The molecules such as the different components of the energy metabolisms and autophagy, coagulations, and interleukin pathways, and they are highly expressed in COVID-19 brain when compared to the control brain. Further, We see the conclusion, and this conclusion was reached after examining the inflammatory characteristics of COVID-19 strain. The high level of CD141, CCR7, and CD11C expression in these monocytes indicate that the parenchyma and perivascular area have activated these molecules. When monocytes and microglia activate, endothelial cells, pericytes, and villi membrane are damaged, and the blood-brain barrier is likely to be impaired by the release of inflammatory mediators. Our findings suggest that IBA1 positive, CD11C positive microglia and invasive monocytes play a role in inflammatory brain injury.